Topic 6.1 is on DNA and RNA structure. The focus for Unit 6 is gene expression, or the process by which genes, segments of DNA, are used by a cell to make a specific protein to carry out a specific function. DNA is the genetic material, aka the molecular instructions that your cells got from your parents to build the proteins that make up you. DNA basically enables the transfer of information from one generation to the next. Before cell division, DNA is packaged into chromosomes made of chromatin, where the DNA is pretty much tightly wound around proteins called histones. Plasmids are circular pieces of DNA that are not packaged into chromosomes and are copied separately. It took some amazing science for us to figure out the structure of DNA, which is, as you may recall, a double helix comprised of two anti-parallel polynucleotide strands bound together by phosphodiester bonds between the sugars and phosphates of the DNA backbone and by hydrogen bonds between the nitrogenous spaces A and T and C and G. And remember, RNA is one polynucleotide strand with U as a base instead of T. 6.2 is all about how DNA gets copied. This is super important if we want life to continue on planet Earth. DNA replication is semi-conservative, meaning that each DNA molecule consists of one template nucleotide strand and one newly synthesized strand that was built based off of the template strand. To begin, the enzyme helicase moves along the molecule and splits open the two DNA strands, while single-strand binding proteins make sure that they stay open. Topoisomerase makes sure that the DNA strands don't supercoil upstream of the replication fork. When the template strands are exposed, an enzyme called primase places an RNA primer on both DNA template strands, which serves as a starting point for DNA polymerase to synthesize new DNA nucleotides that match up with those on the template strands. Now here's the catch. DNA polymerases can only make new strands in the 5' to 3' direction. This means that since DNA is anti-parallel, one new strand is built in the forward direction, but the other strand needs to be built essentially backwards. So only a piece of the new strand can be built at a time. These Okazaki fragments are sealed together by ligase as DNA replication continues all the way down the length of the entire DNA.
6.3 is on transcription and RNA processing. Gene expression is all about how DNA contains a code that your cells use to synthesize specific proteins. But your cells can't just directly use the DNA to do that, and mRNA, a temporary copy of a specific gene, must be used instead. Transcription is the production of mRNA from DNA, and it's kind of like using a quote to support your argument when you're writing a paper. You won't cite a whole book or an article, but just a piece. RNA polymerase is the enzyme that produces a complementary strand of mRNA based off of a DNA template strand by prying open the double helix and adjoining RNA nucleotides together as it slides down the template strand. It begins at the sequence called the promoter and ends at the terminator. Transcription factors are proteins that control whether RNA polymerase will bind. To make sure that the mRNA makes it safely out of the nucleus and into the ribosome, and so that the right part of the gene is translated, the primary mRNA transcript is processed. A 5' GTP cap and a 3' poly A tail are added, and it's spliced, meaning that the unused or non-coding regions of the mRNA are removed and the coding regions are linked together, kind of like editing a video. 6.4 is on translation, which is pretty much what it sounds like. Once mRNA is processed and transported out of the nucleus, it's brought to a ribosome, which is an RNA and protein complex responsible for decoding the A's, C's, G's, and U's in the mRNA to assemble the correct sequence of amino acids in order to make a functional polypeptide. The mRNA kind of gets fed through the two subunits of a ribosome like putting originals into a copy machine. 3 nucleotides of mRNA, or a codon, bind with the ribosome at a time, while another RNA, called tRNA, uses its complementary anticodon to match up with the mRNA and bring a single amino acid to the ribosome. For example, the codon AUG will match with the anticodon UAC, and the tRNA with that anticodon will transfer the amino acid methionine to the ribosome. The ribosome continues reading the mRNA 3 nucleotides at a time, and tRNAs put together a chain of amino acids based on the mRNA code until a stop codon is reached, at which point the amino acid chain is cut off. You can translate codons into the 20 different amino acids yourself by using a codon chart like this one. A bit more to talk about in AP Bio 6.4. So far, this unit has mostly been about the central dogma of molecular biology, which basically states that all living things have DNA as their genetic material, transcribe individual genes into RNA, and then translate that RNA into a chain of amino acids in order to build a specific protein. Things called viruses are basically just capsules of protein and some DNA or RNA that take control of gene expression and replication in living cells so that they can force a cell to make more viruses, spreading amongst once healthy cells. Viruses are technically not living things because they cannot metabolize or reproduce on their own without basically hijacking an actual living thing. There are obviously a lot of notable viruses, but one group that we're going to be talking about here are retroviruses like HIV. Retroviruses use reverse transcriptase to, you guessed it, reverse transcription and its viral RNA and convert it into DNA, which is then incorporated into a host cell's genome. That way, the virus's genes are now part of the cell's genes, which can stay dormant and get copied with the rest of the cell's genome every time the cell decides to divide.
6.5 is on the regulation of gene expression. An organism's genome contains the instructions to build thousands of different proteins, but no cell needs to express every one of those genes, so it's crucial to control which proteins are being made and which ones are not. Prokaryotes control gene expression through the use of operons, where the transcription of one gene is turned on or off by the presence or absence of a repressor protein, which binds itself to a segment of DNA upstream of a transcribed region called the operator. Molecules called inducers essentially remove the repressor so that RNA polymerase can bind to the promoter and transcribe. Eukaryotes, on the other hand, control gene expression in a number of ways. Modification of histones in chromosomes allows the cell to choose which genes to express on a chromosome. Negative and positive transcription factors control the binding of RNA polymerase. The different ways mRNA can be spliced can also affect which genes are expressed, and translation itself can be blocked with the use of microRNAs, which bind to a processed mRNA molecule and don't allow it to enter a ribosome. Lastly, some cells can just block or break down already finished proteins. 6.6 is on gene expression and cell specialization. If you're watching this, you're probably a human being, which means that you are made of trillions of cells, which, when they work in a group, make up tissues. Groups of tissues make up organs, groups of organs make up organ systems, and a group of organ systems makes up you. Each of those biological levels of organization has a specific job to do. Almost all of the cells in your entire body are going to do one job and one job only until they die. In other words, your cells are highly specialized. For example, a neuron in your brain does a completely different job than a red blood cell. The function of a cell is determined by its regulation of gene expression, or which proteins it does or doesn't make. Each one of your cells has a copy of your DNA, but it's not going to use all of it, just like you're not going to use an entire library's worth of information to write one essay. A stem cell, which is a cell that does not have a job yet, like high school students, receives signals that tell it which combinations of genes to express and by extension, what type of cell to become. A neuron is going to express combination of genes to send impulses, while a red blood cell will express genes that allow it to carry oxygen. 6.7 is on mutations, which are random changes to genetic information that have greatly affected the history of life on Earth. Mutations are the ultimate source of genetic variation, as they count for 100% of all new genes. A change in DNA results in a change in a protein, which leads to a change in a phenotype or a trait. When a single nucleotide is changed, it's called a point mutation, which can alter an mRNA codon so that it gets translated into a different amino acid, or sometimes even a stop codon, thereby affecting the protein structure and function, though other times it doesn't change anything. But when an extra nucleotide or two is inserted or deleted, that's a frameshift mutation. This changes the groups of 3 that a ribosome reads, and therefore every codon downstream of that mutation results in amino acids that are different from what they're supposed to be. Since prokaryotes don't do meiosis and reproduce asexually, mutations are their only source of variation. So when bacteria divide, any mutations they have are passed down to their offspring or vertically, but they can also pass them horizontally with plasmids. Plasmids with mutations can be picked up in a bacterium's environment, transferred directly from one bacterium to another, or even passed along by accident through viruses. 6.8 is on biotechnology, specifically the kind where scientists use techniques and machinery to manipulate how DNA works. The forms of genetic engineering discussed in AP Bio all require the use of restriction enzymes, which are enzymes derived from bacteria that scientists use to cut DNA in very specific locations so that genes can be isolated for our own benefit. Bacterial transformation is when the desired gene, like say the gene that codes for human insulin, is inserted into a plasmid, which is then absorbed into cultured bacteria to be expressed in large quantities. If we want billions of copies of a target gene, polymerase chain reactions can be used to control high-speed DNA replication using a special heat-resistant DNA polymerase. Restriction enzymes can also be used to tell if the DNA from one source is the same as the DNA from another source through gel electrophoresis. This involves treating 2 or more samples of DNA with the same restriction enzymes and then using a machine to basically sort out the cut up pieces of DNA by size to create an image that can be analyzed. This is what's used in DNA fingerprinting.